Good afternoon, or good evening, good morning, but it's good afternoon for our speaker. I welcome you at this seminar in the series of the Department of Epidemiology of the Harvard School of Public Health. My name is Bert Hoffman. I'm the chair of epidemiology, and it's my distinct pleasure to invite for a seminar today Dr. Shushi Ogino. I just checked it, 11 years ago that he gave a seminar starting his whole field of molecular pathological epidemiology. And Dr. Ogino has worked on that very diligently over decades and more now. Dr. Ogino is a professor of pathology at Harvard Medical School. He is the American Cancer Society professor. He is the chief of the Molecular Pathological Epidemiology program in the Department of Pathology of the Brigham and Women's Hospital. He is an affiliate member of the Broad Institute of Harvard and MIT, and he is a professor in the Department of Epidemiology in our department. And it's wonderful to have you, and you will talk about I think it is your favorite topic, namely pathology and its marriage with epidemiology. Why are pathobiological analyses and insights valuable in epidemiology? It's my great pleasure, Dr. Ogino, to give you the floor, please. There we are. Thank you.
Okay, great. Thank you so much for kind introduction, Professor Hoffman. Well, anyway, I don't have any conflict with interest at this point, and you can photograph post slide with my credit anytime, anywhere, except for do not post ones. This picture illustrates my interest over more than a decade. So I'm interested in cancer. I'm interested in knowing how the cancer happen, develop over years and progress too. And obviously epidemiology play a significant role in deciphering all the exposures and disease incidence outcome. And then I'm also a pathologist to better understand cancer in the pathological molecular level too. And this is important because good lifestyle can prevent cancer and improve therapy, which is a great impact. But I think it is a little bit relatively understudied. People are interested in drug and patient outcome a lot, but not much on the lifestyle. So it's a great opportunity. And of course, pathological profiling can enhance epidemiology, and I have shown this over more than 20 years now. So I can show you some example too. And to merge these fields, so I name it to molecular pathological epidemiology, which is quite easy to understand. But now really, this integrates many other fields such as immunology, microbiology, data science. So it's getting bigger, and actually, so we decide to put a different name in the meeting series we have running. So the next meeting, International MP meeting, is rebranded to IPPS, Integrative Pathobiology Population Sciences. And this will happen in likely Boston in 2027. So please stay tuned. You can visit mpmeeting.org. That's easy to search too. So back to this picture, and when I made this picture, actually, we didn't think. I mean, obviously, we already know this. We already know. We have known tumor develop over decades. Tumor develop over many years. So the patient come to your clinic, come to attention. But cancer won't happen in one day, 1 month, even 1 year. It's developed over years and decades. That's very important. To address this, I have been very lucky to get involved in these incredible studies run by Harvard investigators. One is a nurses health study, And then the second one is a health professionals follow-up study, and then nurses health study too. Together, about 300, almost 300,000 people, starting follow-up 1976, the first nurses health study, and questionnaire every 2 years, and then biospecimen collected. And then, you know, for epidemiologist audience, I don't need to mention how important it is, but no one knew who would get cancer until it occurred. So that is a very, very important strength of these prospective cohort studies. And I don't need to really emphasize. And then another thing is, this also, I don't need to emphasize how important to get the long-term exposures to in a longitudinal manner, something like this, because tumor develop over decades. So, obviously the Harvard investigators caught many cancer incidents, not only colorectal cancer, focusing on colorectal cancer, but there are many other cancer types. And we all pick up any cancer, you know, occurring in this cohort. Probably number is about 50,000 or more, right? 10, maybe 100,000. But anyway, I'm focusing on colorectal cancer and precancer adenomas. It's about more than 10,000 too. And then I was recruited to Harvard to get involved in the collection effort. So we have about 3,500 tumors across the US. It takes, now it's 24 years I have been here. We are still collecting tissue. And then my lab, we are working on the tumor analysis, including pathology, genomics, epigenetics, and more recently microbiota. I mean, not really recently, you know, we have been analyzing microbiota more than 15 years now. In situ single cell analysis, and now most recently AI spatial biology. I would like to emphasize, obviously this is enormous effort, incredible cohorts, and so many great people I have worked with. And I just put some name, but I really cannot list everybody get involved. But particularly, I think that Dr. Frank Spicer, if he didn't start the nurses health study, I would not be here. And also the Charlie Fuchs, who gave me a job, incredible job. If he were not here, then he had not been here. he left here. But anyway, if he didn't give me a job, I would not be here. So these are really efforts of everybody. So and thank everyone. So anyway, and also over many years, I have been very honored to work with great statisticians here too. And especially Maureen Wang has been leading the development of a statistical method to address the complexity of tumor phenotype into epidemiology. And then Donna Spiegelman, Daniel Nebo, Tyler Van der Weel all help us a lot too. And for example, just one example, we apply inverse probability weighting method to adjust for selection bias. These 10,000 pre-cancer incidents, we have about 3,500 tumor tissue. There is a selection here, so we can adjust for selection bias by IP weighting. So that's kind of just one example, and published in the Bert Hoffman's journal, European Journal of Epidemiology too. So, and also the AI spatial biology is also probably the latest development, but we are actively working in this area and is working with brilliant computational biologist Kun-Shin Yu at the Harvard Medical School and many other colleagues too. So we are trying to understand tumor molecular immune microbial spatial pattern and biological patterns too. But for interest of time, I don't go into this detail. And I just go back to how we can analyze this vast amounts of data. One thing I would like to emphasize is that AI is brilliant and decipher many different patterns in the tissue. And they may also epi data, but AI cannot make decades long cohort follow-up data, exposure data. We really need to follow hundreds of thousands of people. And also we need to have lots of investigators for these efforts. So I would like to emphasize that AI cannot make those data yet. And then, so we follow, this is a nurse, these are nurses health study, health professional follow-up study participants, and then we have a cancer tissue, cancer incidence and outcome. And then among them, we get about 1,700 colorectal cancer tissue available. We can subtype them. And then we try to link exposure to particular subtype, particular pathologic signature, pathologic marks that can improve causal inference too. So that's our approach. Then I'd like to just show you the data based on the pathogenic bacteria in tissue. So those are kind of hot at this point, and also we really get the very interesting results I would like to show you. So 3 bacteria here I picked, and then we analyzed over 15 years, more than 15 years. One is PKS Escherichia, PKS positive Escherichia coli producing Colibactin. And the other one, another one is Fusobacterium nucleatum or Fusobacteria animalis. And then Bacteroides fragilis is another one. And so the most recent, so this is do not pass slide because we haven't published. The very exciting finding is that being breastfed, we asked the participant question whether you are you were breastfed as a child, as a baby, actually. So many of them remember, and I remember too. I'm not the breastfed; I am a formula fed. And then we have follow-up data. We have a cancer incidence, and then we have a bacterial subtype. And actually, we tested this hypothesis whether being breastfed influencing cancer incidence with particular with bacteria. That's our hypothesis testing, and then also we can test whether colorectal cancer with the Colibactin mutation signature, we call it SBS88. It's caused by a Colibactin, which is produced by this PKS positive E. coli. So we can test all these hypotheses in the study. So today is actually the only data slide I can show you here. So I can explain. So we compare not the breastfed, and then yes, so being breastfed. We compare incidence of colorectal cancer for decades, right? 3, 4, 5, 6, 7 decades. And then we see cancer incidence, hazard ratio between these yes and no group. And then we can actually test the hypothesis in a different subtype. So in this case, PKS positive Escherichia coli positive subtype and negative subtype in blue. So the positive subtype group, you can see a big increase in the hazard ratio. This is a minority subtype, but we can actually see a big effect. And then majority subtype, bacterial negative type, we don't see any significant increase. So that's very interesting and exciting. Actually, we actually hypothesized the other way around. So because we had an assumption that breastfeeding is good, it may be protective. Actually, it's not, and it's opposite. Quite interesting. So we check other bacteria too, like a Fusobacterium nucleatum, positive versus negative. A positive and negative subtype. So you can see a high increase in high hazard ratio for Fusobacterium nucleatum positive subtype too. And then compared to that negative subtype, there's no significant increase by breastfeeding. And then lastly, if we Bacteroides fragilis, positive and negative subtype 2, we can see increase in the bacterial or Bacteroides fragilis positive subtype, but not negative subtype. So those three bacteria are independent variable. They don't correlate, well, actually they are really independent bacteria and species, and then they have a different biological meaning. And nonetheless, all three of them increase by breastfeeding. It's very, very fascinating result. And actually we check SBS88, Corybactin mutational signature, caused by, produced by PKS positive E. coli. Corybactin produced by PKS positive E. coli. And then it actually show SBS88 positive subtype increase, although we don't have as many cancer cases, so its association was attenuated, but we see a same trend. So that's interesting. So what's going on? So first of all, right, the exposures decades ago can influence cancer incidence, and then differentially by a bacterial subtype, which is a fascinating result. And that is really telling us tumorigenesis occur over decades. That's why I emphasize this point. And so the breastfeeding increase all of these subtypes. Why? Our hypothesis right now is likely mothers' bad habits, going to milk in some form, maybe bacteria, maybe something else. And maybe formula can protect from that bad effect. But we really more data needed. So we really need to collaborate with perinatal, neonatal, pediatric epidemiologists. I think we really need to break barrier and collaborate more widely, to address all these problems, because tumor develop over decades. We need to know what's going on from early life. We need a life course epidemiology approach. So anyway, and then another thing is, so the traditional approach, you can pick any risk factor, but usually effect size is kind of small, like hazard ratio 1.2, 1.3. Maybe if you are lucky, maybe 1.5 is a good number. However, you can see clearly if we can select the right subtype, you can see a big effect size. So you don't need as many sample size if you can expect a big effect sizes like this. So that's great. So actually we put this in almost 8 years ago. We put the picture here. So you can see if we can pick a right subtype, actually you can see big effect size, even hazard ratio of 2 or more. And then another important thing is this subtype is the preventable subtype, preventable fraction. You can change behavior, lifestyle. In this case, probably mother's lifestyle change can prevent kids' cancer risk decades later, which is fascinating. So that is a good message. So actually I came here 24 years ago. So over 24 years, this approach has worked well. The long-term exposures we get in the cohort studies, and then we capture all the cancer incidents. We get tissue, and we subtype by tumor markers, and then can link to a subtype with exposures to gain more pathogenic insights, as well as the big effect sizes. So this approach worked very well. And then we look back, our first study is the working with Andy Chang and also Charlie Fuchs. We published in this New England Journal paper. So the hypothesis is quite simple. Long-term aspirin use may impact and prevent PTGS2 positive subtype incident. So that PTGS2 is known as cyclooxygenase 2, which is an enzyme inhibited by aspirin. So the aspirin, if you take aspirin, these subtype, depend on the cyclooxygenase 2 activity, can be reduced, incidence reduced. And then we see exactly supporting our hypothesis. So that's why we can publish the New England Journal of Medicine, which is quite a tough journal to be accepted, as you know. And then after 18 years, already 18 years, and then this is still the newest and the only finding, only study. You never find any other study address this question. Long-term aspirin use, decrease incidence of PTGS positive subtype and PTGS negative subtype. There's no other study, no other group could do similar prospective study, which is so amazing, right? 18 years later, still newest study, only study. And then, not just that, we published about 35 studies total after that about the long-term exposures and then tumor incidence and then subtype. And this is Dr. Eva Schonhammer, his colleague, still I think affiliated here too. And she's Vienna now, chair there, the EPI chair, and also Minian Song, our colleague here, of course. And then we published long-term all these exposures and the cancer incidence with some subtype which makes sense to be linked to exposures too. And 35 studies total. I mean, some of the studies others can do, like basic tumor subtype, like a very common one, like microsatellite instability testing. I mean, that's kind of standard test for all cancer type now. But those are kind of replicable by other studies. But many of them, actually, we put all these are not studied by any other group yet. So still up to 18 years, all these are new study and the only study. So I just thought about this fact. I think this is quite a unique approach. This, we measure long-term exposures and the incidence and get tumor tissue and then subtype. That's kind of new approach. It's sort of, or I would say not a new, but it's kind of unique approach. So finally, I put a name on it. So that is PC IBM, not the PC IBM, but it's just happened to be the same name, like the prospective cohort, incident tumor, biobank method. It's a method name. It's a generalizable method name here, which is PCIBM. Can assess long-term exposures and tumor incidence plus characteristics. So the basic idea is, prospective cohort study, longitudinal, typically big enough, like 50,000 or more, follow up for decades, and then, various interval for surveys to collect the information and also the cancer incidence too. I mean, and mortality. And then biospecimens there. And then cancer incidence, and then get the tissue analysis. So all these are kind of not the common approach, I would say. But there are other cohorts did the similar approach. So we just put the name on it so that people can recognize this is kind of unique approach. So we haven't done yet, but our plan is after we publish flagship paper, we like to plan some consortium. It's about, I guess, about 15 prospective cohort studies with the incident tumor tissue bank. And that's our cohort. Actually, nurses 3, and then guts. Also, we try to get tissue too. So start thinking about it. So I put it here, but no tissue yet. But anyway, outside of Harvard, there are multiple cohorts with tissue data, tissue resource available, but not utilized enough, I would say. And then UK Biobank, I don't think they've collected tissue yet, but probably I hope so too. And maybe help with NCI cohort consortium too, because they are prospective cohort consortium. I think tissue collection in these prospective cohorts are clearly precious resource because not many. Just I think about 15, but I may miss some, but probably not more than 20, 30, So that's very rare. And then underused and underappreciated. That's why I didn't know if any other cohorts have tissue. And that's why we now define the PCIBM so that people recognize this is very unique and high value. That's my goal. And then another thing is that over years of all these studies, what we learn from about the risk factors and carcinogenesis. So how do risk factors cause cancer? That's a big question I have for 24 years since I came here. So obviously some risk factors cause genetic, epigenetic changes, such as I mentioned Corybactin, PKS positive E. coli. They can cause Corybactin mutational signatures. And then also Marios Giannakis, and we published that red meat and carnivore as well. I mean, and Ed Jovanovich too. We show red meat, processed meat linked to alkylating mutational signature. And then also alcohol, folate, linked to alignment hypomethylation. I just mentioned work with Eva Schonhammer. So those risk factors exist, causing genetic and epigenetic changes. But many more risk factors seem to cause tissue and microenvironmental changes. So exposures alter tissue microenvironment. And actually something like a smoking, which causes smoking, tobacco signature in lung cancer, upper respiratory cancer, but head and neck cancer, but not in colorectal cancer. In colorectal cancer, we clearly see evidence for changes in tissue microenvironment. And then we look at many other things like diet, aspirin, omega-3 fatty acid, vitamin D. So the omega-3 fatty acid, vitamin D, I show you a paper example working with Mingyan Song and Andy Chan. We see a link to immune cell infiltrate in tissue. And also now breastfeeding is another one. It's a breastfeeding breastfed as a baby is a risk factor for tissue microenvironment decades later, which is really fascinating. And so basically exposure change tissue microenvironment, that change alter the microenvironment can give selective advantage for some mutant clones, because we know every one of us now have mutations in our body, which we call the somatic mosaicism. So we have a mosaic of cells and some mutant cells in cancer driver genes actually. So but many of us are okay. We don't get cancer for life. But there are mutations there, mutants are there. So I think it's important to change lifestyle and then change microenvironment so that they don't give selective advantage. So that's the message. And then we put this idea 10 years ago as an etiologic field effect. So the field effect was originally considered as kind of tissue cellular changes without evidence of malignancy, and in a sense that they now the field effect expanded to epigenetic genetic changes in the cell. Now I think with the microenvironment too, what we call an etiologic field effect. And then also I just recently came up as a new concept named environmental vulnerability. This means some tumors are vulnerable to environment and microenvironment. So if we can change lifestyle, behavior, environment, it can be prevented or treated. If tumors depends on environment and etiologic field. So that's kind of a new concept we hope we can write up. And I mean, if you want to write up with me, please let me know. I haven't started, but hope to start soon and publish maybe next year or two, maybe two. So anyway, so 10 years ago we published this paper with Paul Lockhead, who was a GI gastroenterologist at MGH that time. Anyway, so for example, people smoke in lung, airway, or head and neck, they cause mutational signature, but in colorectum, it causes a microenvironmental change, which give a selective advantage, selection advantage for mutant clone to grow. Some of the mutants have a very high mutational load, like MSI high subtype. So they tend to occur people who smoke. And then they tend to be a multiple too. Synchronous tumors can happen. So this model can explain well how smoking, cigarette, tobacco cause carcinogenesis. So how many time I have? Okay, 10 minutes or so, less than 10 minutes. So I go through a little bit. Early onset cancer is the next topic. So I think I cannot talk, just a 5 minutes talk here. So if you are more interested in, actually, there is a video, 20 minutes or so, is my ESMO keynote lecture, ESMO Congress keynote lecture last year. Some new data this year, but still a big problem, same, not changed. So you can listen and see what's a big problem in this area. And also, we will have a meeting in Italy. So if you're interested in visiting Italy, it's a nice mountainous area, I heard. So that's early onset cancer meeting there too, if you're interested. Early onset cancer is increasing all over the body as well as all over the world. And there are many obesity-related cancer and also GI cancer, digestive cancer, including head and neck also digestive system too. So that means probably diet, microbiome, immunity play key roles. And also, as I mentioned, long-term risk factor exposure is important because the cancer occurring young age, like 30, 40, right? Starting decades ago, meaning, right? Maybe starting a baby or child. That's our hypothesis. And then another important thing is this is not just early onset cancer epidemic. I think that the people get older. Now young people, 30, 40, 50, have an increased cancer risk. If they get older into 60s, 70s, and 80s, we will have many more chronic diseases, including cancer. So that is quite a warning sign. We just see a tip of iceberg now, but we will see whole iceberg data decades later from now. I can warn, And so for lack of time, we don't go detail, but basically what I would like to show you is if we can identify early onset cancer tumor features, something like this, which already replicated, which can give us pathogenic clues, like a Lyman hypomethylation is common in early onset colorectal cancer, as well as immune suppressed microenvironment, and also Corybactin mutational signatures I mentioned. It's caused by Corybactin produced by PKS Escherichia coli I mentioned. So those are replicated finding and feature of early onset colorectal cancer. So actually before, studying early onset cancer, we have all age cancer with those early onset tumor features. Then we can guess these are probably risk factor for early onset cancer. So one example is, again, the alcohol, low folate intake, long-term intake, long-term days associated with the increased risk for Lyme hypomethylated tumor. And then inflammatory diet, no aspirin, increased risk of immunosuppressed CRC incidence. And also Western diet, being breastfed, is a risk factor for PKS Escherichia coli positive CRC. All a long-term exposure data we had. So as you can imagine, no other group could do similar analysis yet. So that's kind of a important value of PCIBM. So to summarize my talk, molecular pathological epidemiology studies give us pathogenic insight, and the PCIBM, prospective cohort incident tumor biobank method based discoveries, many only and US findings still up to 18 years. And also I give you environmental vulnerability concept, which is meaning tumor is vulnerable to environmental lifestyle changes. So those are preventable fraction too, preventable cancer, important and likely treatable too, if they still depend on environment or etiologic field. So that's important clinically and in public health too. And if you let me know if you want to write up this with me soon, And then rising early onset cancer, especially colorectal cancer, is a problem. And then we know our immunosuppressive features, Lyme-1 hypomethylation, Colibactin mutational signatures, features of early onset colorectal cancer. And then we can use all age cancer to decipher etiology of early onset cancer too. And the big question is being breastfed really a risk factor? That's we don't know yet, but we need to test more. We need more data. So I'd like to thank everybody, panelists, everyone. I just mentioned, but key people here. And also, again, I put here, and so I also put up more than 20 years of support. I also highlighted Walter Willett and Meir Stanford, in addition to Frank Spicer and Charlie Fuchs. But all of these people helped me along the way. So thank everybody very much. And then also, of course, the cohort participants, nearly 300,000 people dedicated to providing us with a huge data over longitudinally, long-term exposures, which is enormous volunteerism. And also various US hospital pathology department giving us materials that also enable us to do this incredible study. And the funding, and also lab members too, led by Dr. Guys, now is a PI at the National Cancer Center too. And then funding, major funding here too. And then again, if you're interested, please attend those meetings. One is early onset cancer, the other one is pathobiology in population sciences, molecular pathological epidemiology. Thank you very much for your attention.
Dr. Ogino, Tsuchi, thank you very much for an overview of decades, 2 decades or more even, work on the link between pathology and epidemiology. Highly interesting. May I invite people for making comments or asking questions? And you are okay with us doing a Q&A session, huh, Sushi? Yeah.
Mm-hmm.
Okay. Yeah, we have limited time, as you know, because there's a teaching program after this. But if there are any questions, please either let it know in the chat, or put your hand up, or your whatever, you speak up. If not, let me let me please, can I start with a question? Which I have quite a few actually, but one, this concept of the etiologic field and the etiologic field effect, perhaps together with environmental influences or so. Can you develop that a bit more? To me, it seems an interesting, very interesting concept, but I do not quite have a sense of what you mean by this idea of etiologic field.
I see. So what that means, the etiological factors, risk factors, get into body and then get into tissue where they change many things, but change first probably tissue, extracellular matrix,
Yeah.
microenvironment. They change some way, maybe affecting their local immune cell there, or maybe tissue, bacteria. I mean, we now know. I mean, when I was a medical student, I was taught that the tissue should not have any bacteria, but that was wrong. As of course, we know, in the human body is full of bacteria, even in the, in the tissue microenvironment. So those are changed by environment, and then change the microenvironment by those lifestyle risk factors and environmental exposure. Now, in turn, this microenvironment, may affect clone with a mutant. Some mutant clone can grow faster in some particular environment. So one example we think is, for example, Fusobacterium or Bacteroides. The experimental studies show those are immunosuppressive. They try to defend themselves by blocking immune response against them. Of course, otherwise those bacteria will be killed. So that same mechanism, similar mechanism also work to protect the tumor cell. So tumor cell may not be highly attacked if those bacteria are present. And then eventually the clones grow and then become a tumor. So that's kind of what's going on in the etiologic field. It's just an example of bacteria.
As an example, yeah, because so it's not a very specific type of concept. It's an overall kind of unifying idea, but you
general,
it in depending on the specific risk factors and outcomes. Okay.
Right.
But I mean that is then more specific.
Think about the cancer epidemiology. I think that everybody needs to think about why the risk factor causing cancer, and this is
that brings me to a question. Namely, a very interesting finding, I think, on breastfeeding, and then the relationship between the PKS plus E. coli variant having an odds ratio or a hazard ratio of 2.5 or so with colorectal cancer. And the question is, of course, can you speculate on why that is? How would that work? And so in what way is the etiologic field in this case affected?
That's a big question. Obviously, nobody knows the answer yet, including Minya-san or Marios Giannakis. We are always thinking about why this happened. And I think one thing is, I already put that slide, but the mom's bad behavior make a milk change somewhere. Maybe they already have a PKS E. coli, or I don't know, but the bad milk goes into gastrointestinal tract and they may grow PKS E. coli. We don't know. And then finally, they may cause the mutational damage, or they can just reside in tissue and causing mutational damage for a long time, like decades. But we can still pick the PKS E. coli in tissue after 60, 70, 80 years later, after being breastfed. That is so amazing, fascinating. And then, of course, we can also detect SBS 88 Colibactin mutational signatures too. So it's basically it's still speculative, and we need more studies using child cohort, perinatal
Right.
cohorts for sure.
Right. Well, an interesting thing is when you look at in one of your slides, you look at the early onset cancers on the rise. Yeah, that's of course that's a fascinating and also a bit disconcerting type of observation that we see in, I think, many cancers. The American Cancer Society has particularly focused on that, and I think that's very reasonable. Now, you say at a certain point in one slide, it's the Western diet that is related on the one hand, and then breastfeeding. And I'm not sure. How do you reconcile those two things?
Oh no, I think each of them are kind of is risk factors, and I think maybe mothers eating Western diet and Western lifestyle
Yeah,
is a risk factor itself, and the breastfeeding is a risk factor itself, and then the kids grew up with the Western diet, that is also risk factor itself too. So I think that we like to tease out more, but I think in summary, actually, we found more like glycemic load, insulin resistance,
yeah.
metabolic abnormality
Yeah.
is a driver for this PKS positive E. coli positive cancer. We haven't published that yet, too.
That's very interesting. Yeah, I had heard that. So that's a very interesting observation indeed. Yes. Now, if you think in terms of the breast cancer observation, so the E. coli observation, you said it has not been seen in other studies. Did I get that right? Okay. Is that because it is not being replicated, or is it because people do not have the tissue and the data to replicate it?
Right. That's exactly. So I mentioned about 15 studies with the incident tumor biobank. I mean, 10 excluding us. The issue is pathologists are not epidemiologists. They don't recognize value. They are not dedicating their effort. And then the case numbers are not as big as us. Like 300, it's difficult to do study. About 300 cases, you need more than 500 at least to do this type of study, and also lack of funding too. So I think we can make a consortium, a PCIBM consortium, to revitalize all these precious, resource. And I hope of UK biobanks, they should start collecting tissue as soon as possible. I hope, they are really incredible data set too.
You may also look at the so-called German national cohort, which is 250,000. It's a bit smaller, but it's a very large. And I think they have tissue there, and there are pathologists working in that. So that's a good link for you.
Right, great.
Yeah.
But I must say, Netherlands cohort study.
Say it again.
Netherlands cohort study is a pioneer.
That's another one. Yes, that's another one. That's like
they started long before us, and then they published before us. First paper is 2003.
Yeah, that is true. That is also.
So really, I appreciate they are really pioneer.
Thank you very, very much, Dr. Ogino, for linking pathology, molecular pathology in particular, with epidemiology. And I'm quite sure we will have very exciting findings in the next decades that you work and your colleagues and pathologists work together with epidemiologists. Thank you very, very much.